Hello! Do you ever wonder why do enzymes react differently? But first, let's define enzymes. Enzymes are proteins that help speed up chemical reactions in our bodies. They are essential for digestion, liver function, and much more. Too much or too little of a certain enzyme can cause health problems. Moreover, enzymes in our body can also help healthcare providers check for injuries and diseases. Enzymes build some substances and break others down. And you must remember that all living things produce enzymes, and they are also found everywhere in nature. Now, we will further explore and deepen our knowledge about enzymes in our third experiment. In this task, we will encounter the enzymes present in varied food sources such as apple, pineapple, potato, and milk. Also, after this experiment, we will distinguish the roles of these enzymes, the factors affecting them, and how they are involved in biological functions. Before doing the experiment, it is important to wear personal protective equipment to ensure your safety. Now, we are ready to perform the experiment. Hello guys! I am Shidal Alison Ercilia Francisco, and I am here to present to you the task 1 of model number 1 for experiment number 3, which is entitled Juice Cook and Dry. For the first procedure, first, peel the whole apple and chop it into small pieces. After chopping the apple into small pieces, prepare the cooking pan and then open the stove and set it in low fire. Add 1 glass of water to the pan together with the chopped apple and then cover it for 20 minutes or until the apple looks tender and soft. Since the apple was already tender and soft after 20 minutes, I smash it using a fork until it became a puree. After that, I transferred the puree to a bowl-like container with a cover and set it aside to do the second procedure. For the second procedure, I measured 1 cup of meat tenderizer. After that, I also added a cup of water to the jar and mixed them together to form the needed solution. Let's prepare a container and put 3 tablespoons of the apple puree that we made on the first procedure. After that, pour 1 cup of the solution that we made in the procedure 2 and mix it thoroughly. After mixing, filter it using a fine strainer. Let's leave the solution for 10 minutes. After 10 minutes, there's a formation of white foamy structure in the surface of the filtrate. There are also visible small bubbles present within the foamy structure, thus making it looks like the foamy structure in a hot creamy coffee. And if we are going to compare the results in procedure 1 and 3, in procedure 1, it is observable that after chopping the apple, it immediately turned brown. And this enzymatic browning is not unique to apples. Moreover, after cooking the chopped apple for 20 minutes, it immediately ripened. This is because temperature is one factor affecting enzyme activity. Compared to the third procedure, we added a mixture of meat tenderizer and water to the puree, allowing the formation of a foamy white substance and small bubbles in the surface of the filtrate.
Hi, I am your experiment master Crizel, and at this point we will now proceed to our task 2, what keeps them from browning?
First is to prepare 10 cubes of apple of the same size. Second, prepare and label the covered containers. Number 1 for control, number 2 for water, number 3 for oil, number 4 for lemon juice, and number 5 for onion. Third is to place 2 cubes of apples in each container. Then, put the different substances on the surfaces of each apple cube. Use a spoon to put water on the surface of the apple slice. The same goes for the oil and lemon juice. Lastly, keep all inside the refrigerator overnight. As we can observe, the different treatments of apple cubes vary in terms of browning. If we were to rank them 1 to 5, where 1 is that with extensive browning, the order would be number 1 for the apple cubes with water, number 2 for the control, number 3 for the apples with oil, number 4 for the apple cubes with onion slices, and number 5 for the ones with lemon juice. The rate of browning differs because the substances in the treatments have significant effects on the enzymatic browning of apples by inhibiting PPO activity.
Good day, lab enthusiasts. I am Gigi, and today I will be performing the bubbling veggies experiment.
Put 1/4 cup of hydrogen peroxide in 4 separate plastic cups. After that, put a cube of potato in 1 cup, a cube of onion in the 2nd cup, a cube of apple in the 3rd cup, and a cube of pineapple in the 4th cup. Compare the 3 cubes in terms of the bubbles. The potato produced the most bubbles among the 4. It sunk in the bottom and has a strong reaction with the hydrogen peroxide, causing it to produce more bubbles than the rest of the materials. The onion produced bubbles but not as many as the potato. It can also react with the hydrogen peroxide but not as strong as the potato, thus it can produce lesser bubbles. The onion seems to be floating and its bubbles had adapted the shape of the container. The apple, on the other hand, can produce minimal bubbles. It has a little reaction with the hydrogen peroxide, while the pineapple cannot produce any bubbles when reacted with the hydrogen peroxide.
Hello everyone, I'm CJ Andrade, and today we're going to do task 4, which is entitled Magic Bubbles.
First, I prepared 4 plastic cups. I then put tape on it and labeled it from 1 to 4. After that, I put 5 teaspoons of vinegar in cup 1. For cup 2, I first made the baking soda solution. I put 1 teaspoon of baking powder in 150 ml of water and stirred it thoroughly. And then, I put 5 teaspoons of that baking soda solution into cup 2. I then put 1 strip of potato in cup 1. I did the same for cup 2. I let it soak for 5 minutes. While waiting for the strips of potato to be soaked for 5 minutes, I set it aside first, and then I readied cups 3 and 4. I then put 5 teaspoons of hydrogen peroxide per cup. After 5 minutes, I transferred the strip of potato from cup 1 to cup 3. I also transferred the strip of potato from cup 2 to cup 4. After that, I observed the reaction that would take place. For the strip of potato that was transferred from cup 1 to cup 3, there was no reaction that occurred. However, for the strip of potato that was transferred from cup 2 to cup 4, there was a reaction that took place. We can observe bubbling on the potato.
Hello Science World, it's me Claudette, and this time I will be demonstrating the last task entitled Clot for Milk.
First, prepare 2 glasses of 100 ml fresh milk. Add 1 teaspoon of pineapple juice to the first glass. Add also 1 teaspoon of heated or warmed pineapple juice to the second glass. After that, stir the solutions and record for the results in 5 minutes. After 5 minutes, the glass of milk with heated pineapple has no effect. The surface of the milk is just as plain as it is. On the other hand, the other glass with milk plus pineapple juice results in bitter tasting milk. You can also see a dotted orange structure on the surface of the milk and somehow clotted or curdled milk. This is due to the effect of the pineapple bromelain on the milk.
Now, you have witnessed how enzymes work and react differently in our third experiment. I hope that you learned something from this experiment and realized the significance of enzymes not only in our body but to all chemical reactions. To wrap it up, we recognize the roles of enzymes from varied food sources. More so, we learned that several factors can affect the rate of which enzymatic reaction proceeds. And these are temperature, pH, enzyme concentration, substrate concentration, and the presence of any inhibitors or activators. Definitely, enzymes are essential to help speed up chemical reactions not only in our bodies but to all living things to balance out the buildup and breakdown of different substances.